Upstate Medical University in Syracuse, New York, invites you to be the informed patient with the podcast that features experts from Central New York's only academic medical center. I'm your host, Amber Smith. Why are older adults more vulnerable to serious flu infections? And could we strengthen the body's natural lung defenses? A researcher from Upstate is exploring these questions, and he's here to discuss his work. Dr. Mobin Karimi is an assistant professor of microbiology and immunology at Upstate. Welcome to the Informed Patient, Dr. Karimi.
Thank you. Nice to meet you.
You were awarded funding recently from the National Institutes of Health for research looking at how certain immune cells protect the lungs from viral infection, particularly in older adults. Can you tell us more about these immune cells?
So these immune cells are developed in a very early stages of embryonic development, basically before a human body is formed. They're very infancy, their development, and they keep developing to the first trimester, second trimester, and the third trimesters. During the third trimester, the cells from the livers where they were born, they migrate to the bone. And from the bone, before the birth, they go to lungs and different tissues, and they become resident cells. Basically, they live there for the entire life, and they don't move around. So they are there for the first line of defense. So in the science, we call it innate lymphocytes. You're born with it, you're hardwired in your system where you get it from mom and dad. So basically, those are the cells that you don't acquire it, you don't do vaccination, you don't do anything else. You are born with it, and everything is hardwired in your systems. And these cells are local. Basically, if they go to lung, they will stay in the lung. If they would go to kidney, they will stay in the kidney. Or any other body part, skin, they stay there for the rest of their life. So they are the first line of defense system. Basically, what the scientists knew so far, that they are like a repair engineer. Basically, lungs are the first exposure to the outside world, the things we breathe, the things we encounter, the harmful things we do. And what this cells does is they repair the tear and wears when we encounter from outside.
So let me make sure I understand this. They are born in the liver and they develop in the bone, and then they get to the lungs, and that's where they stay.
You're right. They develop in the fetal livers before there's a bone, and then they migrate to the bone before the baby is born. And then within the third trimester, they go to different organs. They're not only resident of the lungs, they're resident in many organs of your body.
So the ones you're studying are the ones that go to the lungs, but these immune cells go to different organs in the body.
You're absolutely right. They go to different organs of the body. The reason we were interested in it is because for a long time we thought asthma is the disease of the old age. But as you know, that's not true. Young people can also develop asthma, so is older people. Asthma is associated with obesity more than age. So we were interested to figure out what this cells does during asthma infection. So basically, this cells make asthma worsen. Because what happened is during the asthma, your smooth muscle is contracted, they become really tight. And that tightness, the cells produce more inflammatory cytokines because their main job, as I said, is repairing. When they produce more cytokines per cells, and those cytokines cause more inflammation, and your smooth muscle is more contracted, it becomes more tighter, so it worsens the asthma. But our interest is what will be the other functions of the cells. So diseases like influenza virus, like we got common flu, we get COVID, those are all respiratory infections. So these cells, before us, other people have studied it, they know that they do the cleanup job. Once there is infection, there's a lot of damage. There's a lot of damage to the lung area. So what this cells does is they clean it up. But we have discovered that this cells has a dual function. They can convert to another population called ILC1. So ILC is innate lymphocytes. They're ILC1 and ILC2 and ILC3. We were interested in ILC2 because they're pretty much resident cells, but we discovered that they can go back to become ILC1, not only to clean the lung and damage, but also fight infection. And they can fight infection more effectively than other cells.
Now, do these particular cells last a lifetime, or do we get new ones as we age, or are these the ones that are present at birth? Do they stay with us throughout life?
That's an excellent question. A lot of time we know that our immune system, our immune cells, they generated from stem cells, and they can last a day, a week, a couple of weeks, and all of this. But the resident cells, they stay in your lung for the entire life. But how do they regenerate themselves? Because scientists now believe that the stem cells, the mother cells that give rise to the cells, they migrate to the the organ where they can reside for the rest of their life, so they can reproduce within the organ. Scientists have shown over and over again that the cells does not migrate from one organ to another. They do not go back to the circulation, because when we take a mice or a human blood, we find very, like almost none of them in the circulation, but we can find in the resident tissues. But they can stay for almost 75 years. But what happened is that over the period of time, it is not only specific to the cells, but the entire immune systems get weakened, and these cells become more prone to exhaustion, and they can also reduce the numbers as well.
This is Upstate's The Informed Patient Podcast. I'm your host, Amber Smith. I'm talking with Upstate researcher Dr. Mobin Karimi about his study of lung immunity in aging adults. Your research focuses on a particular pathway. Can you explain what this pathway does?
Sure. This is the base for our grant, and this is the base for our several grants. We have just published a paper in JCI Insight where we shown that there's a molecule called beta-catenin. Beta-catenin is a molecule that stay in the cells, and from the cells, they go to the nucleus where they can interact with the specific genes and tell that specific gene what to make. So we made a mouse model that will overexpress or stabilize the population called beta-catenin. And those stabilized beta-catenin mice produce a lot of regulatory T cells. And those regulatory T cells are able to protect lung from lung hemorrhages, which is the major cause of autoimmunity, lupus, and many other autoimmunity models. When you probably heard or seen patient with autoimmunity, they cough blood. So there is significant damage in the lung, and the red blood cells go to the lung. So its inflammatory cells go to the lung and cause damage in the lung. So what beta-catenin does is they increase the regulatory cells, and those are not just ordinary regulatory cells. They are special regulatory cells. They express different molecules. So they are the best to repairing it. So while we are publishing this paper, we also discover that mice with stabilizing expressions of beta-catenin can also express a lot of innate immune cells, which is called ILCs. So our goal in this is to figure out if the beta-catenin specifically regulate ILC. In this grant, we propose to knock out beta-catenin specifically on ILC, or overexpress it in ILC. And how does that impact health? Because one can design, or there's already existing drug that you can use it to stabilize beta-catenin. But in one caveat, that beta-catenin is a growth hormone. we are born with the growth hormone, is also part of the innate system. But what happens is when people have cancer, cancer will target that specific growth hormone called beta-catenin. So right now, when you talk to people with the cancer research, beta-catenin is the major target because what happened, cancer hijack that pathway to use it for their own, for the tumor cell growth. But this pathway is also critically important for the development of immune cells. We have shown, and other people have shown, that if you don't have that beta-catenin, you knock it out, or you don't have it in the mice, there will be no T cells, no immune cells will be developed. So those are critical molecules. So what we have done it, we either pharmacologically or genetically overexpress that beta-catenin, and we have enhanced ILC2 population, which is all mostly the affect the innate lymphocytes. And those are the cells that when we challenge those mice with the flu virus, even a 2-year-old mice, which is equal to 80-year-old human, they can clear the virus. But a normal mice, they cannot clear because ILC decrease, immune cells are decreased. They basically die from the viral infection. That's why we, in the people in the clinic and research, develop special vaccine for people with age, because also we have shown that beta-catenin specifically decreases with age. So the decrease of beta-catenin impact the ILCs and also resulted it cannot fight virus.
So you already know that it has the capacity to improve the immune response.
Yes, we do have that showing in the mouse model that when we overexpress or stabilize express, even 2-year-old mice can fight the virus. And the other things we are currently working, these mice never get fat. No matter what we give them, we can give them Western diet. When we give a Western diet to a normal mice, a small mice become the size of a rat, and obesity comes with almost, they call it the mother of all disease, autoimmunity, inflammation, not only in your body, but also neurological inflammation. So these mice that stabilize beta-catenin expression, they don't become obese, and they don't develop asthma as well.
Do you think you'll be able to answer the question about why older adults are more vulnerable to serious flu infections?
Like I said, one of the critical factors is we have to understand that the growth hormones are decreases with age. And why does it decrease with age? We don't know yet, and we try to figure out what makes them decrease. So if we can enhance those growth hormone back into the people body, that will enable them to fight virus, respiratory infections. But again, people have to change their lifestyle as well. Healthy food, healthy environment, and the growth hormone is really critical for immune balances, this thing called homeostasis, where the immune cells will be happy and less inflammatory. But we are only focused on beta-catenin. There are several growth hormones that other researchers and other scientists have shown that they decreases with age, and that wear tears cause normal inflammations. If you take a 75-year-old person, it will have more inflammation of swelling and exhausted cells than a younger person. And when we look at the molecular mechanism, beta-catenin and all of the immune systems, they get decreased significantly. So our hope is that if we can use pharmacological approaches very specifically targeted those immune cells and those hormones, that they will enhance the production and eventually they can fight the virus and have less inflammatory response.
So it seems like you believe we'll be able to strengthen the body's natural lung defenses. Do you have any predictions for a timetable?
It is very difficult to predict on the timetable. Number one, we do experiment with the mice, and mice has very short span of life compared to human. I'm pretty sure that we can generate pharmacological approaches, drug development that can strengthen older adults, that they will have a quality of life and they can fight viruses more effectively by improving their immune system, mostly their innate immune system where they are born with, because over 70 to 80 years, there decreases. And if we can pharmacologically support and enhance those protein, they will be able to have a better quality of life. So we cannot enhance people aging. Nobody can do that. That would be impossible. But you can improve the quality of life. A 75-year-old person have the ability to be independently walk, go to the grocery stores, play golf, visit the doctors, and the quality of life are more important than prolonging the age. So for the quality of life, we want to figure out what specific pathway can be targeted that we can improve that quality of life.
Well, it's good to know. Thank you so much for taking time to tell us about your work, Dr. Karimi.
Thank you for having me. And it's always nice to talk to you and see the people in our community benefit from our work.
My guest has been Dr. Mobin Karimi, an assistant professor of microbiology and immunology at Upstate. The Informed Patient is a podcast covering health, science, and medicine, brought to you by Upstate Medical University in Syracuse, New York, and produced by Jim Howe, with sound engineering by Bill Brokel and graphic design by Dan Cameron. Find our archive of previous episodes at upstate.edu/informed. If you enjoyed this episode, please invite a friend to listen. You can also rate and review the Informed Patient podcast on Apple Podcasts, YouTube, or wherever you tune in. This is your host, Amber Smith, thanking you for listening.